Welcome back from the break. We're on the home stretch now. Moving into session 4. Session 4 will be on the scientific challenges and advancements of long-acting injectables. Our session leads include Dr. Lucy Fang, currently serves as the Deputy Director of the Division of Quantitative Methods and Modeling in OGD's Office of Research and Standards. Prior to that, she served as an Associate Director and Team Lead of the Quantitative Clinical Pharmacology team within DQMM. We also welcome a session lead, Dr. Bing Lee, who serves as the Associate Director for Science for the Office of Bioequivalence in OGD. Our first presentation in session 4 is on Q1 and Q2 challenges from a BE assessment perspective. Our presenter, Dr. Daping Cui, joined FDA in 2015 and currently is an acting team lead in OGD Orange Book. Our second presentation is on the application of quantitative modeling and simulations to BE determination for long-acting injectables, sharing research progress and regulatory experience. Our presenter, Dr. Kevin Fang, joined the Quantitative Clinical Pharmacology team at DQMM RS OGD in April 2019. Our third presentation is on the mechanistic modeling of complex injectables, recommendations to navigate regulatory challenges. Our presenter, Dr. Kandakar Alam, is currently working as a senior staff fellow at the Division of Quantitative Methods and Modeling in the Office of Research and Standards. His role in the division is to utilize translational tools such as physiologically based pharmacokinetic modeling to address specific questions pertinent to drug development process and/or regulatory decision making. Our final presentation in this session is on the recommendation of partial area under the curve metrics and product-specific guidances for long-acting injectable drug products. Our presenter, Dr. Sharon Thomas, joined the RAS DQMM in 2020, where she contributes to research involving population pharmacokinetic modeling and physiologically based pharmacokinetic modeling focused on bioequivalence and in vitro and vivo considerations. Please join me in welcoming our first presenter, Dr. Daping Cui.
Good afternoon, everyone. My name is Daping Cui. I work in the CEDAR OGD Office of Bioequivalence, Division of Bioequivalence 1. Today, I'm going to present the topic of in vivo pharmacokinetic bioequivalence studies for long-acting injectable LAIs. In this presentation, I will discuss some considerations and challenges in the PKB study for this complex dosage form, especially from the perspective of a BE reviewer. The first disclaimer, my presentation reflects the view of the presenter and should not be construed to represent FDA's view or policies. So for the outline of my presentation, I will first give a brief background of long-acting injectable LAI. I will then go over the BE study recommendations. After that, I will discuss some of the general considerations in the study design of in vivo PK studies for the demonstration of BE of LAI product. I will also discuss some common challenges in the conduct of in vivo PK study for LAI. And due to these challenges, we typically identify multiple deficiencies during the review of PKB study submitted in the NDA application for LAI product. I'm going to share some of common deficiencies. And in the end, I will conclude with some summary and key takeaway message. So one special characteristic of LAI product is that this dosage form provides sustained and continuous release of drug substance over a period from days to months when administered via intramuscular, subcutaneous, epidural, and intra-articular routes. So this release characteristics make LAI treatment a preferred option for some therapeutic areas because it improves patient compliance and treatment adherence by reducing dosing frequency through maintenance of plasma drug concentration over a longer time period compared to many other dosage forms. So LAI have been developed and are approved for the use in several therapeutic areas of chronic disease, especially as antipsychotic, hormonal therapy, and treatment of substance abuse disorders. However, due to the challenge in the development of LAI, especially in the formulation development and in vivo B study, to date for majority of innovative LAI, there are no generic product approved through the 505 pathway. There is also a regulatory requirement which contribute to the complexity of generic LAI development. According to 21 CFR 3-1494, in general, a drug product intended for parenteral use must contain the same inactive ingredient and in the same concentration as the reference listed drug RLD, with the exception that differences in preservative, buffer, and antioxidant may be allowed if appropriately justified by the applicant. So this regulation should be taken into consideration in the generic LAI development. Based on the different formulation strategies, LAI product could be characterized into three groups: polymer-based, for example, PLGA or non-PLGA based system, the oil-based delivery system, and drug crystal suspension. A typical PK property of LAI product is the so-called flip-flop concentration time profile. So following injection of LAI, a large portion of the injected product initially remained in the dosage form in the injection area. The absorption rate from the injection site is slower than the elimination rate from the systemic circulation. Therefore, the long apparent half-life of LAI actually reflects the rate of absorption, and the time to steady state is also a function of the absorption rate. So depending on the specific LAI drug product, one of the approaches listed in this slide may be recommended to demonstrate BE between test and the reference LAI product. So for some LAI drug product, an in vivo PK study is recommended to demonstrate BE. And for some LAI product, BE is demonstrated using in vitro studies in combination with one in vivo B study. This may include quantitatively the Q1, Q2 sameness, comparative in vitro physicochemical Q3 characterization, the comparative in vitro drug release testing, and an in vivo B study. The third type of B recommendation is that in vitro study used as an alternative to an in vivo B study based on a totality of evidence approach, where the B of a Q1, Q2 generic formulation may be established with comparable Q3 characteristics and a comparable in vitro drug release profile. The agency has published product-specific guidance, PSGs, with B recommendation for many LAI products to facilitate generic drug development. The examples listed in the table of this slide represent the three recommended B approach which I presented in the previous slide. So for medroxyprogesterone acetate injection suspension, BE should be demonstrated with in vivo single-dose parallel B study with a PK endpoint. For a risperidone injection, the applicant is recommended to provide information of Q1, Q2 sameness, the physical-chemical characterization of PLGA polymer, the in vitro drug release testing, and an in vivo crossover steady-state PK study to demonstrate BE. While for penicillin G benzazine injection suspension, the applicant has two options to demonstrate BE. One is the in vitro option, which includes the information of Q1, Q2 sameness, Q3 characterization, and in vitro drug release testing. And the other option is the in vivo approach, which is to conduct a single-dose parallel PKB study. So as a sensitive, accurate, and reproducible approach, the in vivo PK study is recommended for demonstrate BE for many LAI products. I will briefly go over the study design and the challenges of PK study in the next couple of slides. So this slide shows the flow chart of the general consideration in the study design. So briefly, if there's no safety concern for healthy volunteers, the BE study can be conducted in healthy general population. And for this situation, typically a single-dose PK study is recommended. If there are safety issues to administer a LAI product in healthy volunteer, then patient population should be used for the BE study. And depending on the dosing regimen of the specific LAI product, either a single-dose PK study or multiple-dose steady-state study is recommended. To continue with the consideration of study design from several aspects. Due to the long apparent half-life, generally a parallel design is used for single-dose PK study of LAI, considering the time needed for complete washout. And for steady-state PK study, parallel or crossover design may be recommended. Regarding the strength used in PKB study, in general, the highest strength is recommended unless there's a safety concern. PK study may also be recommended for additional strengths. For example, when the formulation of multiple strengths are not proportionally similar. And for certain long-acting injectable, for example, a risperidone injection, any strength could be chosen for the BE study, given that all strengths are from the same bulk and there's no additional safety concern. Regarding PK metrics calculated from PK study, generally Cmax, AUC last, AUC infinity, and Tmax are calculated for a single-dose study, whereas for a multiple-dose steady-state study, the steady-state parameters are also required, including AUC tau, Cmean, and fluctuation. For certain LAI product, based on the clinical relevance and the formulation characteristics, partial AUC are recommended in addition to AUC last AUC infinity. For example, for naltrexone ER injectable suspension, it is recommended to include AUC 1 to 10 hour and AUC 10 to, it's recommended to include AUC 1 to 10 days and AUC 10 to 28 days to account for the multiphasic release profile and therapeutic threshold. And among all these parameters, only Cmax and AUC parameters are subject to the 90% confidence interval evaluation. So another consideration in the study design is a muscle injection site for LAIs administered via the intramuscular route. For certain LAI products, based on the ROD information, for example, in the labeling, the both gluteal and deltoid site should be included in the study design for adequate site representation. However, typically demonstration of BE at each of the injection sites may not be necessary. As example, this recommendation is included in the PSG for paliperidone palmitate ER injectable suspension and medroxyprogesterone acid injection suspension. Another consideration is steady state. A multiple dose steady state PK study is recommended for some long-acting injectables. For example, antipsychotic LAI. For this type of study, it should be noted that sufficient number of doses should be administered to achieve steady state, and the applicant should evaluate steady state based on the trough concentration data. So the complexity of long-acting injectable formulation, the release characteristics, and the specific BE recommendation pose many challenges in the conduct of PKB study for this group of product. For example, the requirement of certain patient population and their existing treatment regimen cause significant recruiting difficulty. And another challenge is that the long half-life and the required time to achieve steady state resulted in very long study duration. And another issue associated with the patient study with long duration is the high dropout rate. So high variability is one of the major issues for parallel study, which will require very large sample size. And variability may be contributed by multiple factors, for example, demographics, clinical center effect, and injection site. And other challenges in the PK study of LAI product include steady state determination, safety concern, especially those related to serious adverse event. And finally, the multiple center study with long study duration also pose challenge in retaining the required number of reserve samples. So next I will share some example deficiencies identified in the submitted NDAs for long-acting injectable. One is about carryover effect. In some applications for the crossover study, steady-state PK study, the applicant did not demonstrate there was sufficient number of doses between treatment, which means insufficient washout before the intensive PK sampling. Therefore, cannot adequately characterize the steady-state PK profile in the second treatment period due to the carryover. There were many deficiencies related to the high dropout. For example, the number of subjects not balanced in terms of injection site, gluteal and deltoid, or imbalanced subject number in terms of treatment test versus reference due to a high dropout rate. And applicant did not evaluate the impact of this imbalance on the study outcome, or did not pre-specify how to evaluate this imbalance in the study protocol. The AE, adverse event and safety is another area we typically identify deficiencies for PK study on LAI. The difference in AE profile poses concern on the safety of test product, especially when there are more serious adverse events following test treatment compared to the reference treatment, and the applicant did not provide sufficient details and justification for the different SAE profile. Many deficiencies were also related to the concomitant medications. The co-med are commonly used in patient study with long duration. In some submitted PK studies, the applicant did not demonstrate the lack of interference from the concomitant medication on the analytical method that is used to measure the analyte. Or the applicant did not provide sufficient evaluation of the in vivo PK interaction between the study drug and the co-med, which are likely mediated by, for example, the P450 enzyme pathways or drug transporter. And lastly, another type of deficiency for multiple center clinical center study is that applicant did not provide evaluation of the clinical center effect on the BE statistical analysis, or did not pre-specify the analysis in its protocol and statistical analysis plan. So with all the discussed consideration and challenge in the PKB study for long-acting injectables, this slide showed summary and some takeaway message, which I think both the generic industry and the agency should consider, and how could we work together to facilitate the generic LAI development and to improve the approval rate. So overall, B study design for LAI should account for the formulation design, for example, the release controlling mechanism, and also the dosing regimen, dosing frequency, and the study population. For steady state PK study, appropriate number of doses to achieve steady state should be determined and balanced by the need to minimize study duration. The dropout rate should be taken into consideration in sample size estimation as well as statistical analysis. There should be appropriate PK sampling scheme to accurately capture PK parameters, especially for those with partial EOC metrics. Applicants should conduct sufficient pre-study method validation to examine the interference of concomitant medication and also evaluate the impact of potential in vivo drug-drug interaction. The appropriate safety monitoring should be included in the PK study. And finally, applicants should take appropriate statistical approach for evaluation of clinical center effect and injection side effect as needed. And this approach should be pre-specified. I would like to thank my colleague for the comment and effort to help in the preparation of this presentation, including Dr. Bing Li in Office by Equivalent, Immediate Office, and Dr. Qing Liu, Dr. Wu Pomanshi, Dr. Zhen Zhang from DB1. I also like to thank the comment and suggestion from my colleague, either currently or previously worked in the Office of Research and Standard, Dr. Liang Zhao, Dr. Yan Wang, and Dr. Satish Sharon. Thank you very much. I'm looking forward to the discussion in the panel discussion session.
Hello, here is Kevin Feng from Quantitative Clinical Pharmacology team from DQMM ORS. Today I'm going to present the topic of application of quantitative modeling and simulations to bioequivalence determination for long-acting injectables for sharing research progress and regulatory experience. Here is a general disclaimer. This presentation reflects the views of the presenter and should not be construed to present FDA's views or policies. Here is the outline of the presentation. To understand challenges in pharmacokinetics bioequivalence studies for long-acting injectable drugs, provide opportunities with model integrating B approach via GDUFA research program, collaboratively advancing the field with industry and other shareholders, while FDA regulatory experience in pre-ENDA meetings. Then we have conclusion discussion for future directions. Let's look at the challenges in B studies for LAI products. So for normal B study, it normally will take a few days to a few weeks. However, for a LAI-B study, it will take a few months to a few years. The problem is because a slow release, but it's not elimination. It has a very long half-life. With this very long half-life, there's a challenge in patient recruitment. Also, there has a high dropout rates, and also that need to attainment of steady state for LAI products. So another challenge like formulation release challenges that also was listed in the publication below. Here we have examples of FDA-approved LAI drug products and approved ANDAs. We list all of the LAI products here, and in looking at the product, they have a different dose frequency, at different dose durations, some of them a few weeks, some of them a few months. And only 2 drugs have been approved for the ANDA approval. So one is a medroxyprogesterone, the other one is the paliperidone for monthly. So in here, let's look at why there's a really very few drugs have been approved. Here we have the chart to show the challenges associated with different types of LAI-B studies. The 2 categories, one is if it is safe to dose healthy volunteers, then you will dose healthy volunteer with a single dose parallel study. But if not safe to dose healthy volunteer, then it's going to be in patient with multiple dose and either parallel or crossover study at steady state. So the challenges is obviously, so the first one is with a lower power because of parallel study, but with higher variabilities. But for the inpatient, because the steady state, the duration is really not practical very long and have high dropout rate, and also increase the variations. So for the single dose like the medroxyprogesterone, because it do it in healthy volunteer, that's an approved drug. And for the antipsychotic, because they had to do it in patient like paliperidone, paliperidone, risperidone, so there's really long durations. That's not a practical. So here are the challenges for these patient and healthy volunteers for the LAI products. The opportunities with the modeling is really to address some of the main issues in the in vivo B study. One is to shorten the treatment duration. The second one is whether the possible using smaller sample size. So here list of the challenges or requests from the this year's Generic Drug Science and Research Initiative Workshop. Here list of all of the industry share information. The first one is modeling and analysis proposed including in pre-ANDA submission often result in information request. The second one is specific expectation for model integrating evidence are lacking. The next one is provide clarity on extent of validation criteria necessary for models to replace in vivo B studies. For example, population PK models, PBPK models. Development of in vitro relevant predictive methodology for complex drug products. For example, inhalation LAI ophthalmics to be used to provide model input. Along with other regulatory agencies on acceptance of MIDD. And last one is single-dose B study in healthy volunteers instead of multiple-dose B study in patients. Over the last few years, FDA has already funded a few grants and contracts using the modeling and simulation for LAI products. For example, the first one is University of Utah, and then the Simulation Plus, and then Uppsala University and University of Connecticut. So all of the research have been funded and is some of them is ongoing and some of them is complete. And I'm going to share you with what has been proposed, what has been found in the research. Here is a GDUFA research developed model integrating B approach. This is what proposed by ACOP, presented by Andy Hooker 2019. It including how the model building, how to including the uncertainty and using the simulation, then use simulated data for the conclusion by providing the PK endpoint using CMAX and AUCs. In the model, they also including the formulation difference with the treatment effect or sequence effect in the absorption parameters. By using the model integrating evidence framework that we provided in previous slide, here is one of the opportunities that is possible to reduce the study durations. The study using crossover steady-state studies, the full duration from crossover is to doing the crossover between the reference and then test. So there is a possible that if I can do one period using the switchover study and then avoid to including the rest of the study to reduce the studies. So by giving this framework and using the model information that can using the providing the B metric and then given the steady state confidence interval as a normal B limits using 80 to 125%, and then looking at the switch over steady state and the switch over study, then can revise the confidence interval, provide adjusted B criteria like AUC and CMAX, a little bit narrower than the 80 to 25%, that will provide opportunity to using shorter B studies. Here is another opportunity by using the model integrating evidence framework. By making the model, is it possible to identify some of the covariates like the age or sex or other parameters that can be included into the model. For example, some of the parallel studies at the statistical model, after including those covariates into the model, that is potentially increase the power of the study. Those research was proposed by GDUFA research grant and fundings. And here I'm going to share some of the regulatory experience from FDA, especially from the pre-ENDA meetings. Here is one of the examples that the applicant proposed an M&S approach for B decision by recruiting smaller sample size in a pivotal study. So this approach is acceptable, but the detailed modeling analysis plan should be submitted for the assessment. Another example is the applicant proposed a population PK model which was developed from literatures. Simulation was performed to assess the potential impact of missing PK samples because of COVID-19 interruption. Some of the PK sample is missing for the long-acting injectables. The proposal is not acceptable because the applicant proposed using the literature model for the time and error evaluation, and we encourage the PPK model should be developed using actual clinical data and fully validated and capable to determine the formulation difference and be used for imputation to investigate the impact on missing PK samples. After the applicant makes some judgment on the modeling, then we see that approach is acceptable by using the modeling and simulation to adjust the missing PK samples. So this is one of the examples that have been using modeling and simulation and provide information about the missing PK samples. Here is another example that the applicant submitted a population PK model using modeling and simulation approach and provide a modeling analysis plan. The analysis plan is considered reasonable. However, further justification needed to be provided with more details. The last example I want to provide is the applicant proposed a mechanistic erosion-based PBPK model which incorporates in vitro and in vivo data with the associated scientific literature for a virtual BE simulation. The approach is considered insufficient because the mechanistic PBPK model should be able to detect potential formulation difference with proper estimation for type 1 and type 2 errors. The comments on details of modeling strategy was provided for the applicant. The model integrating evidence is rapidly evolving. So here is the common deficiencies. The first one, the applicant did not submit a model analysis plan. The second one is the time error result were based on literature model. The applicant did not evaluate the time error before the virtual BE simulation. The overall modeling approach is lack of supporting materials for modeling process. The model is not able to detect potential formulation difference between test and formulation product. The sample size of virtual BE simulation is a lot larger than the sample size of clinical BE study for model building without sufficient justifications. The last one is the applicant did not understand that the model building and validation in BE decision is more stringent than the POP-PK modeling in new drug development. So here I summarize some of the key components for the modeling and simulation that FDA will consider for the model integrated evidence. For the first two one is a model building and model validations. So for model building and model validations, so on the top one, they normally will require in the POP-PK guidance, so including all of the variabilities, and then the numerical validation and graphical diagnostic. So for the next of two differences from the POP-PK is for the B model, people had to including the formulation difference in the model, for example, including treatment effects or sequence of piercing effect on absorption parameters, and for the validation, the additional validation for the PK metric, including Cmax and AUC infinity, and observed within the simulation like 50% or 90% for both of TNR or PRS actuals. So there's additional validation, and also the model need to include formulation difference. The third one is once you have the formulation difference, then you be able to looking at the time and error and type 2 errors. And for the time and error is really to looking at whether your model is able to detect a formulation difference, and whether your model is for the power and sample size have sufficient power and sample size. And then after you have the model validation, and then you be able to do the simulation, including all the metrics, including like AUC, CMAX, AUC infinities, and using all of the different methods, including the model-based BE, model averaging, and bootstrap selections, model-informed methods. So that's the kind of simulation that you'll be able to look at. Finally, then you have the model decisions for the BE, the including the different types of data sources and model uncertainty, and then the BE matrix, especially for the 90% confidence interval for the PTR ratios for all the PK metric that's referred into 80 to 125%. So those are the key components that FDA will looking at. And here, so after a few slides, we are going to look at some of the challenging questions. The question 1, which of the following statement is true? A, applicant can propose a model integrating evidence as an alternative approach without detailed model building and validation process plan. The second one, the model should be able to detect the sensitivity of formulation difference and provide confidence in simulation for BE decisions. Third one is, applicant can use a smaller sample size in pivotal study and use M&S for virtual BE simulation without considering uncertainty. The answer is the second one, B. The model should be able to detect the sensitivity of the formulation difference and provide confidence in simulation for BE decisions. The second challenge question is, which of the following statements is not true? Due to the COVID-19 situation, modeling and simulation approach can be used for BE decision by recruiting smaller sample size in pivotal study, providing with detailed modeling analysis plan. B, due to the COVID-19 public health emergency, a large number of PK samples was missing. A population PK model can be developed using actual clinical data. The fully validation be capable to detect the formulation difference and be used to imputation to investigate the impacts of missing PK sample. C, applicant can propose a mechanistic erosion-based PK model which incorporates in vitro and in vivo data with the associated scientific literature for a virtual BE simulation without providing the sensitivity of detecting formulation difference. So the answer is C, is not true, because the applicant for the PBPK model, they still need to be able to detect the sensitivity of the formulation difference, so that we be able to looking at your model, that your model is validated for the difference between the test and reference products. So here is the conclusion. We list a few possible model integrating evidence approach using M&S for LAI BE assessments that was proposed by recent FDA-funded grant, Contrast for Research Institute. It's including the model simulation to conclude BE, using model integrating BE, conventional model averaging, and bootstrap model selections. And then we also show the research on the model to inform BE statistical model, including switch study for adjustment of BE limits, and also including the covariance effect for the parallel study. FDA is committed to advancing this area and has been providing guidance to industry-wide pre-ENDA meetings and incorporating the feedback from key shareholders via public workshops. FDA encourages innovation M&S approaches to overcome the challenge of BE assessment, but detailed modeling analysis plan should be submitted for the evaluation. So for those modeling evaluation, that you can refer for the key components slides. Finally, we have the acknowledgements that include the DQMM, Mion, Yong, Yi Gong, and Quantitative Clinical Pharmacology team, and Lucy Fan and Liang Zhao. And also appreciate for the OIS, OIO, Rob and Lei, and also the other grants and funding from the University of Uppsala, Andy Hookers, Xiaomei Chen, McCausens, and many more. Thank you. For any of the questions, we can discuss in the panel discussions. Thank you so much.
Hello everyone, my name is Khandokar Alam. I work in the Division of Quantitative Methods and Modeling under the Office of Research and Standards within Office of Generic Drugs. Over the last few years, we have seen increased interest from generic drug developers to utilize mechanistic modeling on complex injectables in support of their proposed alternative bioequivalence approach. In my talk today, I'll be providing some recommendations to navigate regulatory challenges in this regard. Disclaimer of my presentation. Couple of learning objectives from my talk today. I'll be briefly talking about the utility of mechanistic modeling in the development of complex injectables. Then I will talk key considerations for model-based alternative bioequivalence approach. Then I will share my views on how to navigate regulatory challenges with 3 hypothetical scenarios. Lastly, I will be discussing key consideration in preparing pre-NDA meeting package. There are different types of complex injectables. Here I have listed few common types. First one is long-acting injectables or LAI suspensions, which is often injected into muscle or subcutaneous tissue, and the drug is released from the injection site for a prolonged period of time. Polymer-based implants and in situ forming implants is another important class where the drug is generally released from polymer matrix due to hydrolytic degradation of the polymer. Lipid-based nanoparticles such as liposome for injection often designed to circulate in systemic compartment for a longer period of time and generally deliver the drug to the target tissue. The last one I have listed here is IV colloidal solution or nanoparticle suspensions, which involves complex manufacturing process such as nanotechnology. Many complex injectables, including LAI suspensions, have no or limited numbers of generic drug products. When it comes to conduct an in vivo BE study, each type of complex injectables has its own challenges. Some of these challenges include increased variation in PK parameters. However, the source of PK variability often remains vague. Another challenge is to recruit patients for the clinical study, as the healthy subjects are not often recommended. High dropout rate of patients, as many of the clinical studies are few weeks to several months long, often not practical to perform single dose BE study. Mechanistic physiologically based pharmacokinetic modeling or PBK modeling has the potential to save time and resources in the development of complex injectables. For example, PBK modeling may support drug approval via virtual bioequivalence study, may help us to understand the effect of key formulation attributes on the systemic exposure, such as the effect of particle size, surface morphology of APIs, pH or viscosity of the formulation on the systemic PK profile. We can also use PBK model to define safe space for critical formulation attributes. PBK modeling can also be utilized to explain potential source of PK variability. For example, PBK model can integrate the interaction of API with physiology, for example, local immune cells, and PBK model can account in vivo aggregation of particles at injection depots from muscular, intramuscular, or subcutaneous injection. With the help of PBK model, we can also establish mechanistic IVIVC. We can extrapolate IVIVC from animal model to human subjects by accounting specific physiological differences. Key considerations for model-based alternative bioequivalence approach. We should take extra care for developing the model. Model should describe the relevant physiology, integrate key formulation attributes, and account the interplay between formulation attributes and physiological parameters. If the model is well built, the model verification task becomes easier. Primary model verification should focus on ensuring predictive ability of the model for the drug product. Secondary model verification step is required to ensure the creditability of general PBPK model structure by verifying the model against wide range of drug products administered through same route. It is very critical to identify the application of the model. Model can be used to conduct virtual bioequivalence study or establish mechanistic IVIVC or explain source of PK variability or other purposes. There is a white paper from FDA authors where we have discussed the dermal PBK model. You may utilize relevant ideas from this paper in the development and verification of complex injectable PBK model. In next few slides, I'll be discussing 3 hypothetical drug development scenarios to give you more clarity on how to overcome regulatory challenges when PBK modeling is part of your alternative bioequivalence approach. First example is on LAI suspension for IM use, and this type of drug is often used by patients for a prolonged period of time. Let's consider a hypothetical PSG. It is recommending in vivo BE study with PK endpoint in patients who are already receiving a stable regimen. Your product development team may be thinking that PHGs of azacitidine and triamcinolone acetonide has the options for in vitro approach to establish BE. So why don't you propose FDA for in vitro characterization based BE approach for these hypothetical LAI suspensions? Based on my regulatory experience, my thinking is the recommendations and rationale are specific to a drug product. You cannot really translate one PSG recommendation to another drug product. Another important thing to notice here is that patients do not use azacitidine or triamcinolone acetonide injection for a prolonged period of time. So the potential safety and efficacy risk associated with this hypothetical LAI suspension may be much higher compared to other drug product. If the in vitro characterization based BE approach is not a viable option, and if there are certain challenges in conducting in vivo BE study and you want to avoid those challenges, you may think of using model-based alternative BE approach. Let's assume that innovator company of this drug product has established IVIVC and some relevant information is available in the NDA review. You want to use this information to develop a PBK model-based IVIVR, and to reduce your drug development cost and time, your IVIVR is based on data of only one formulation that has intermediate release rate. Now you have proposed to predict the bioavailability of the test and reference drug products by utilizing PBK modeling and IVIVR and ask the agency for their feedback. So the question is, how would we consider your proposal? Of course, we have to review your proposal first, but I can share my thinking based on these hypothetical scenarios. We have to understand that IVIVC is generally established by knowing the manufacturing history and intentionally formulating batches with various release rates such as slow, medium, and fast. However, your proposed IVIVR is based on one formulation that has intermediate release rate. NDA applicant established IVIVC really cannot be adapted by generic applicants for supporting regulatory approval. However, you can use this knowledge as a starting point to develop the IVIVC for your own test formulation. Relevant question: can you use animal model to establish IVIVC for test formulation? Based on my understanding, if a generic applicant want to use IVIVC to support the approval of the test formulation, only human data should be applied for regulatory consideration. Animal data cannot be used in lieu of human studies for bioequivalence determination. However, results from animal studies can be used for product development, such as identifying CQS of a drug product or to justify the use of proposed alternative bioequivalence approach. Example 2 is on colloidal solution or nanoparticle suspension, and this product is administered intravenously. A hypothetical PSG recommends both in vivo BE study with PK endpoint and in vitro studies like particle size distribution and physicochemical characterizations. Since the product is administered intravenously, can in vitro characterization based approach be explored for this drug product, considering the API of this drug product is dissociated from excipients upon intravenous administration? Here is my thought. Though the product is administered intravenously, it is not a simple IV solution for injection. The product is manufactured utilizing nanotechnology. Thus, variation in manufacturing condition may result some differences in surface chemistry of the API, which may influence the plasma protein binding of the API and thus can lead to difference in systemic drug disposition of the API. If a generic applicant wants to avoid in vivo BE study for whatever reason, model-based approach can be a good alternative. Let's discuss a hypothetical drug development scenario utilizing modeling approach. Can the data from tissue distribution studies in mice with the test and reference drug product be used to develop a PVK model? Second question is, can the mouse model be extrapolated to human and the model be used to conduct virtual bioequivalence study to establish BE? To answer the first question, it is your choice how you want to develop your PVK model. However, when it comes to extrapolate the mouse model to human, we have to be little cautious. API of nanoparticles can be substrate of transporter proteins or CYP enzymes. There could be certain differences in the disposition characteristics of API between mice and human. For example, human orthologs of certain transport proteins are absent in mice. Major metabolizing enzyme formed in the human could be different than that formed in the animal model. Plasma protein binding of API in human and mice may differ substantially, and API may eliminate substantially faster in one species compared to others. So extrapolation of mouse PVK model to human PVK model is not a straightforward process. Simple allometric scaling may not apply here. However, the approach may be viable if you develop a mechanistic model which can account specific differences in physiology. If you want to utilize model-based approach, your proposal should have sufficient description regarding the PVK model development and predictive ability of the model. Your proposal should discuss how the formulation attributes of the test and reference formulation would be incorporated into the model. You should also discuss your plan on estimating potential formulation differences and virtual bioequivalence analysis, such as reasonable consideration of inter and intra-subject variability, number of subjects, type 1 and type 2 error analysis. The third and last hypothetical scenario I'll be discussing today is PLGA-based implant of API. The release of drug from PLGA matrix is quite complex. Molar ratio of lactic and glycolic acid in PLGA polymer, molecular weight of PLGA are assumed to be critical in the degradation of PLGA and subsequent drug release from the matrix. Many PLGA-based drug product also shows distinct PK profile, for example, primary and secondary peak, which is assumed due to complex interplay between formulation sequence, implant specific properties, physiologies, etc. Let's assume a hypothetical PSG which recommends single dose parallel in vivo BE study in patient with peak endpoint. We already know some of the challenges of in vivo BE study. In this case, crossover BE study may also pose some challenges as the drug may potentially show some carryover between subsequent periods due to the long-acting nature of the drug product. So to overcome these challenges, model-based approach may be utilized. Here is a hypothetical question, and the proposal in this example is a bit different. Here a generic drug development company wants to conduct an abbreviated in vivo parallel BE study and wants to use the PK data from the in vivo study to generate virtual subjects in PVK model. The proposal is to conduct PVK model based virtual bioequivalence. We can consider this as a hybrid virtual bioequivalence approach. A mechanistic PVK model can be utilized to demonstrate virtual bioequivalence given that the model is adequately describing the complex drug release mechanism. The model is informed with formulation sequence, API specific physicochemical properties, implant specific properties. The primary PVK model is adequately verified for its predictive ability. For example, you can intentionally manufacture variant formulations by changing PLGA molar ratio, molecular weight, drug loading, porosity, etc., and assess the model's predictive ability against those formulations. The PVK model is verified against a wide range of similar drug products to ensure the creditability of the model, and the plan to identify potential formulation difference and virtual bioequivalence analysis is adequately described. If you are planning to utilize model-based alternative bioequivalence approach, I would encourage to interact with the agency early. Your model doesn't have to be completed, but it should be in a sufficient state of development to allow for scientific discussion of specific aspects of the model. Key considerations in preparing pre-under meeting package. You should clearly state the purpose of the modeling approach. Any modeling assumption that has the potential to mitigate any actual difference between test and reference formulation would be more suspect and need a greater level of justification. When the model is proposed for establishing virtual bioequivalence, you should describe your plan adequately regarding how potential formulation difference would be estimated. You should clearly state the study design and data analysis plan for the virtual bioequivalence. We understand that incorporation of inter and intra-subject variability is often challenging. However, reasonable assumption on variability with proper justification should be discussed. You should also provide consideration on how the virtual bioequivalence approach would ensure acceptable type 1 error by demonstrating the sensitivity of the approach to detect non-bioequivalent scenarios. That is all from me. Take home message from my presentation today. Mechanistic PBK modeling has the potential to overcome challenges associated with in vivo BA study and thereby can save time and resources in generic drug development. Animal studies cannot be used in lieu of human studies for B determinations. However, generic applicants may use the results of animal studies for product development, such as identifying CQAs or supporting justification for the proposed alternative approach. ND applicant established IVIVC cannot be adopted by generic applicants for supporting regulatory approval. Generic applicant may use this knowledge as a starting point to develop the mechanistic IVIVC for their own test product. Early interaction with the agency is encouraged for model-based alternative bioequivalence approach to get agency's feedback on the proposed approach. My acknowledgement to my FDA colleagues. I am grateful for their collaborations and suggestions. If you have any questions on my presentation, please feel free to reach out to me at the email ID listed here. Now, time for challenge questions. Question 1. How can mechanistic modeling be utilized in the development of generic complex injectables? Mechanistic modeling of complex injectable A can help in overcoming challenges associated with in vivo BA study. B can help in understanding the effect of key formulation attributes on systemic PK exposure. C can help in explaining the source of PK variability, for example, by accounting the interaction of API with immune cells, in vivo aggregation of particles at injection depot. D can help in drug development process via virtual bioequivalence study. E, all of the above. Answer is all of the above. Challenge question 2. What considerations should be taken into account during the preparation of pre-NDA meeting package when model-based alternative bioequivalence approach is proposed? Options are A. Clearly state the purpose of the modeling approach and provide relevant descriptions and analysis. B. Provide sufficient information on model development process. C. More justification is needed for model assumption that has the potential to mitigate actual differences between two formulations. D. Provide sufficient evidence and or plan for the model verification process. E. All of the above. The answer is E. All of the above. Thank you everyone for listening.
Good evening, everyone. Welcome to the last presentation of the day. Today, I'm going to talk about recommendation of partial area under the curve metrics in product-specific guidances for long-acting injectable drug products. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. Learning objectives for today. First, to describe formulations for long-acting injectables with partial AUC recommendation. Second, to understand scientific rationale for partial AUC recommendation. And lastly, to describe a couple of case examples. Long-acting drug products for injection, implantation, and insertion are formulated to achieve sustained drug release and action for extended time. For the sake of this presentation, we are going to focus on injectable drug products. Long-acting injectable products are classified into three groups. First, drug substance suspensions. Second, suspensions containing weight-controlling inactive ingredients like parenteral microspheres. Third, polymeric in situ forming gels or implants. Polymer microspheres are control-release systems that consist of polymeric materials that encapsulate the API in a dispersion or as an API core surrounded by a polymer shell. These are multiphasic release systems, showing an initial burst release with the release of API adsorbed to the particle surface or near the surface driven by diffusion. This is followed by a lag phase, which is the time required for polymer degradation and erosion to occur as a result of the release media penetration and polymeric matrix swelling. Then we have an extended release phase where the polymer degrades enough to become water soluble. The polymeric microspheres will undergo mass loss and matrix erosion, continuously releasing the API until depletion. Each of the above release phases can result in distinct phases and peaks in the PK profile for the long-acting injectable drug product. In situ forming implants or gels are injectable liquid dosage form which consists of drug, water-insoluble biodegradable copolymer, and biocompatible organic solvent to dissolve the polymer and deliver the drug in the form of solution or suspension. Upon injection, the aqueous body fluid triggers a phase separation and polymer precipitation, leaving a biodegradable solid depot. The system is also a multiphasic release system, showing an initial burst release caused by the phase separation as a solid depot is formed. This is followed by a diffusion facilitated release phase and a degradation facilitated release phase. Each of these phases are shown in the figure below. What more is partial area under the curve for assessing BE for long-acting injectable? Long-acting injectable drug products have complex drug release mechanisms. Drug substance suspensions show diffusion control release. Polymeric systems show a combination of several mechanisms such as diffusion, erosion, etc., as we saw in the previous two slides. When a single dose in vivo BE study can be recommended, the traditional metrics of AUC and Cmax may not be sufficient to capture the potential impact of formulation and/or manufacturing parameters on the drug release kinetics in vivo, or to ensure comparable drug absorption or exposure for some long-acting injectable drug products. The table here shows several PhDs that have recommended partial area under the curve for long-acting injectables. The examples include leuprolide acetate products, triptorelin, buprenorphine, octreotide, and naltrexone. The clinical design here shows a single-dose BE study, and the formulations involve polymeric systems such as microspheres and in situ gel systems. In the subsequent slides, we'll look at a couple of case examples. Case 1 is leuprolide and triptorelin. Leuprolide and triptorelin are gonadotropin-releasing hormone agonists. The partial AUC recommendation is for day 7 to T. Day 7 to T represents the sustained release phase or plateau phase of the PK profile. The PK curve of the drug product Eligard is characterized by two main phases which include an initial burst release of the drug product followed by a sustained release plateau phase for several days. The initial burst release from Eligard formulation is completed within the first 2 to 3 days. The plateau phase lasts from 1 to 6 months based on strength. So what are the reasons for partial area under the curve recommendation? Once the serum level of testosterone is less than 50 nanogram, the PK profile does not impact the PD result. The PK curve is characterized by a large initial peak due to the high burst release, which constitutes a significant portion of the total area under the curve. By day 7, this initial release is completed. The sustained release portion of the PK profile from day 7 to T is the most relevant period to compare rate and extent of the absorption of the drug release from the test and the reference products. Mean testosterone levels increase above the baseline during the first week following the initial injection and declining thereafter to baseline levels. Therefore, we see a transient increase in clinical signs and symptoms of puberty during the first few weeks of therapy or after subsequent doses. Now, this is consistent across all GnRH agonists. This suggests the importance of the plasma concentrations at the start of the therapy Hence, AUC day 7 to T parameter is clinically relevant based on this mode of action, and hence the sustained release portion impacts the clinical performance of the formulation. Case 2 is buprenorphine. Buprenorphine is a partial opioid agonist used in the treatment of opioid addiction. The partial AUC recommendation is week 3 to 4. Partial AUC here represents the terminal portion of the plateau phase of the PK curve. Here, partial AUC is recommended in the U of Seaman as BE criteria. The buprenorphine atrogel delivery system allows for extended release by the formation of in situ gel. The PK curve for the drug product is characterized by two main phases, which includes an initial release of the drug product, characterized by a single peak. The initial burst release is small and does not constitute a significant portion of the total area under the curve. This is followed by a sustained release plateau phase for several days. So why do we recommend partial AUC for buprenorphine? The exposure response analysis shows a trend of drug liking reduction with increasing buprenorphine exposure. A comparable minimum plasma concentration for potential generic formulation to the RLD during the plateau phase is important. Cmin and the shape of the PK curve should be monitored in the end stage of the plateau phase, such that the Cmin of the generic product would not be less than the concentration of the RLD at the same time point. Due to difficulties of proper characterization and BE assessment on Cmin because of the high intrasubject variability, partial area under the curve from 3 to 4 weeks is recommended in lieu of the Cmin. Case 3, octreotide and naltrexone. Octreotide is a somatostatin analog, partial AUC recommendation of day 0 to 28 and 28 to 56. Naltrexone is an opioid antagonist with partial AUC recommendation of day 1 to 10 and 10 to 28. Here the reason for partial AUC recommendation is because of the multiple peaks in the PK profile. Octreotide, the single-dose PK profile can be seen to the right. The single-dose profile is characterized by a transient initial peak within 1 hour after administration, progressively declining over the following 3 to 5 days, then slowly increasing and reaching a plateau about 2 to 3 weeks. Since the dosing interval is 28 days, exposure from 0 to 28 days is important and hence is recommended as a partial AUC. Octreotide levels continue to increase even after 28 days. Equivalence in exposure during the time interval from 28 to 56 days will help to ensure similarity in steady-state profiles, and hence the second AUC recommendation of 28 to 56 days. Naltrexone single-dose profile can be seen to the right. The single-dose profile is characterized by a transient initial peak which occurs approximately 2 hours after injection. This is followed by a second peak approximately 2 days later and a slow decline approximately 14 days post-dose. Days 1 to 10 captures the large second peak in PK profile. Drug release from days 1 to 10 constitutes a significant portion of the area under the curve. Days 10 to 28 captures the sustained release phase of the PK profile. Prolonged concentration below 1 nanogram per ml is not clinically efficacious for alcohol dependence. The above two partial AUCs ensure similar rate and extent of release between the proposed generic product and the RLD. The regulatory considerations for the use of partial area under the curve for long-acting injectable is clinical relevance. Example, the sustained clinical effect resulting from the control of API plasma concentration within a therapeutic window for an extended duration, such as in the case of buprenorphine. Second, formulation of the dosage form. Example, the complex PK shape resulting from the multiphasic release. The time window relevant to match the plasma profile must be considered, such as in the case of leuprolide acetate. Challenge question number 1. Which of the following long-acting injectable formulations has partial area under the curve recommendation in their PSG? Leuprolide acetate, medroxyprogesterone, olanzapine, penicillin G. Let's take a few seconds to think about the right answer. The correct answer is leuprolide acetate. Challenge question number 2. Which of the following is the reason for partial area under the curve of 3 to 4 week recommendation for buprenorphine long-acting injectable? Comparable plateau phase, identical initial burst release, comparable Seaman during plateau phase, multiple peaks in peaking curve. Again, let's take a few seconds to think about the right answer. The correct answer is comparable Seaman during plateau phase. To summarize, long-acting injectable products have complex multiphasic drug release mechanism. When a single dose in vivo BE study can be recommended, partial area under the curve can be considered, as traditional metrics of AUC and Cmax may not be sufficient to ensure comparable drug absorption and exposure. Clinical relevance of API plasma concentration and the multiphasic release from drug products are the major regulatory considerations for partial area under the curve recommendation. Lastly, I would like to thank my colleagues from DQMM and members of DTP1 and members of Partial Area Under the Curve Working Group for their contribution towards this presentation. Thank you, and that concludes my presentation. I'll be back to answer questions on the panel.
Thank you all for the wonderful presentations. We're heading into our last Q&A panel for today, and if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We'd like to welcome to the Q&A panel Dr. Hao Zhu, who is the Acting Director at the Division of Pharmacometrics, Office of Clinical Pharmacology, CDER. He joined FDA as a pharmacometrics reviewer more than 15 years ago. Dr. Zhu has been a clinical pharmacology team leader for more than 6 years and a QT/IRT scientific lead for 2 years. Then he became the Deputy Director at the Division of Pharmacometrics. And joining us on the Q&A panel is our session leads, Dr. Lucy Fang and Dr. Bing Li. Looks like we have some questions coming in right now, and our first few questions will be going to Dr. Daping Kui, and here is the first question, and this will be a combination with Dr. Daping Kui and Dr. Lucy Fang. Here's the question. In a steady state study, it is required to show steady state attainment of each patient. If any patient does not demonstrate steady state, whether his or her data should be excluded from the statistical evaluation.
Thank you. Thank you very much for the question. It's a great question. First of all, I like to say that the recommendation is the purpose of the recommendation is to demonstrate the bioequivalence of the test and the reference and the steady state condition. Therefore, it is recommended to demonstrate bioequivalence demonstrate that a steady state is achieved or approached for both product. It should be noted that the time to achieve or approach steady state may not be the same for the reference product and the generic product due to the different formulation or manufacture or the physical chemical property. Therefore, this is why the language is provided in some of the PSG. I do understand that there are multiple statistical approach to evaluate steady state. For example, the population-based evaluation or individual subject-based evaluation. And for the individual subject based approach, there should be some factors be considered. For example, if you use a linear regression-based approach, you should also consider the number of trough samples you use or the absolute concentration level. They may contribute to your conclusion of whether steady state is achieved for individual subject. So as a summary, I would suggest that the applicant to first of all to pre-specify the approach to evaluate the steady state in your protocol and statistical analysis plan, and if possible to discuss your approach with the agency, for example, in control correspondence or pre-NDA meeting, and also in the actual statistical analysis to present the result of both, including or excluding the individuals that you consider not achieve a steady state. So the above is my personal recommendation. I would also recommend my colleagues from other discipline to chime in. Thanks.
Thank you, Dapong. Just to extend what Dapong just said, Dapong actually shared with you our current practice in terms of the PACE steady state B study in patients. We do acknowledge that sometimes it can be pretty challenging to conduct steady state B studies in patients, particularly for non-acting injectable products. So we hopefully from today's session, you get a strong message that FDA is open to alternative approaches, such as model integrated evidence approach, where you probably can use a model to correct or subtract the drug exposure from the previous treatment, or you can use model to perform some in silico simulation of what's happening at steady state. And if you do plan to use those model integrated evidence approach, we strongly encourage you to talk with FDA and before you implement such an approach. Thank you.
Thank you both for responding to that question. We do have another question for Dr. Daping Kui, and here is the question. In addition to half-life, what other factors should be considered for PK sampling for in vivo PK study on long-acting injectables?
Thank you very much. This is another great question. Yes, so half-life is definitely the critical parameter need to be considered for the PK sampling design. There are other considerations for the long-acting injectable product. I will give a few examples. For the first consideration is the sensitivity of the analytical method you have established for the BE study. So in combination with the clinical pharmacology, with the PK profile of the study product, you want to ensure your analytical method is able to capture the absorption elimination phase and avoid unnecessary PK sample. Another consideration is a slow release of the LAI. So in general, the Tmax of individual subjects is usually in a wide range. Therefore, frequent sampling around a certain time is not needed. So it's unlike to some other dosage form. Another consideration is that for some other dosage form with a long half-life, which is due to the slow metabolism and elimination, The applicant sometimes can use a suitably truncated AUC approach to characterize the total exposure. However, this approach may not be appropriate for a long-acting product because this approach should, if you choose to use this approach, it should cover the complete absorption phase. But for many long-acting product, the apparent flip-flop kinetics, PK kinetics is due to the slow release of the API from the dosage form injection site. So the truncation of sampling time, the AUC, may not be appropriate. Another consideration as one of the presentation in this section is that for certain non-injectable product, the partial AOC is recommended based on the multiphysics release profile and the clinical relevance. Therefore, the PK sampling should be designed to capture this multiphysics release profile. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Kevin Fang, and here is the first question. Covariate adjustment can also be done for the traditional ABE testing using the GLM or mixed model, which will also improve power. What's the difference of covariate adjustment in a model-based method from that in the traditional ABE method?
Thank you for the question. So we do not normally allow any covariates to be added in the traditional ABE method. However, in our BE guidance, it shows that if you can provide sufficient justification, then the covariates can be added. But for the model-based approach is one of the methods that you can justify to see whether the covariates has a significant on the model, then you can use that covariates for judgment for the covariates. So that's one of the methods that can be done in the parallel study with highly variable drug. So that's the difference between the traditional ABE and the model-based approach. Thank you.
Thank you for responding to that question. We do have another question for Dr. Fang, and here is the question. Can I directly use the model published for the type 1 era evaluation?
Yeah, thank you. Thank you for the question. We normally don't allow that you take the public available model for the data because the data can be different. So that you can use the clinical data to validate the model and then use that model for the Taiwan error calculation in order to detect those formulation sensitivity in your real clinical study. So for the model validation and model estimation, that you can refer to the key components slide that normally people were missing some of the components that we are looking at. Thank you.
Thank you for responding to that question. Moving on to our next panelist. You have a couple of questions that came in for Dr. Khandakar Alam. And here is the first question for Dr. Alam. Can FDA publish a paper on virtual BE study that is based on a model? We understand the benefits, but who better than the FDA to carry the torch and publish one?
Thank you for the question. So this is a nice proposal. So we are actually publishing papers continuously. It is either coming from FDA authors or from our external research collaborator. So keep eyes on those papers. And we also understand that the challenges with the virtual bioequivalence. For example, we know that there are certain challenges for in vivo BA study for each specific drug product. So same challenges also remain for the virtual bioequivalence, because it is very difficult to sometimes characterize the intra-subject variability, particularly in PBPK model, and there are lack of literature sources as well, and we have lack of understanding how to incorporate those variabilities. So for virtual bioequivalence, for crossover study design, we particularly look into the risk assessment, for example, the type 1 error analysis. So I think that any reasonable assumption for the inter-subject variability and reasonable analysis or acceptable optimum analysis for the type 1 error should be considered. And thank you for the proposal. We'll be working with our external collaborator and other internal collaborator as well to publish those papers on virtual bioequivalence.
Thank you for responding to that question. We do have another question for Dr. Alam, and here is the question. You mentioned that long-acting injectables and other complex injectables often show a secondary peak in their PK profiles. Is it absolutely necessary to capture those secondary peaks with PBPK model? Is it possible that the observed secondary peak is just an artifact?
Yeah, thank you for this question. Yeah, this is a very nice question. It is possible that the secondary peak is just an artifact, but we need to look into the PK profile for the individual subjects, and if all the subjects are showing secondary peaks in their PK profiles, it might give us some insights that a secondary peak is likely a true phenomenon. So many long-acting injectable suspensions and complex injectables show secondary peak. We know that, for example, secondary peak in PLJ-based drug product may be due to slower degradation of PLJ matrix and subsequent due to formation of fibrous capsule as a consequence of API and immune cell interaction, and the drug may release from that capsule or envelope slowly to the systemic circulation. So that way we can see some secondary peak. Or so if we are using PBK model,
Dr. Lamas is the moderator. We're losing your reception. We're not hearing your sound at this time. What we can do is we can move on to the next panelist and then loop back around to you so you can just provide a review, just slight summary of what you're saying right now. Moving on to our next panelist, we've got a few questions in for Dr. Sharon Thomas, and here's the first question. Should we recommend a therapeutic threshold, Cmin or C-trough, rather than PAUC?
Thank you for that question. So my understanding is that there are some difficulties in the proper characterization and B assessment on Cmin due to the high intra-subject variability, since Cmin is generally tends to be more patient-specific. So if we look at the later part of the partial AUC, the later part of the PK curve, so the later PAUC is more cumulative PK parameter at the terminal phase. So we would prefer that more than the Cmin. Thank you.
Thank you for responding to that question. We do have another question for Dr. Thomas, and here is the question. Why are partial AUCs recommended for B evaluation of parenteral microsphere formulations?
Thank you for that question. So in case of parenteral microsphere formulation, these are multiphasic release systems. So we want to ensure comparable performance for the sustained release portion. So in the example of leuprolide acetate, you can see that the PK shows a high burst, which is followed by a sustained release. And this high burst constitutes a significant portion of the AUC. So in cases like this, the traditional metrics such as total AUC will not be able to capture the differences in the PK shape adequately, and hence we would recommend a partial AUC specifically for the sustained release portion. Thank you.
Thank you for responding to that question. Moving on to the next panelist, we received a question in for Dr. Hao Zhu. And here's the question for Dr. Zhu. How can the clinical pharmacology programs for the reference product be used to support BE evaluation for the generic long-acting injectables?
Well, thank you. I think generally speaking, clinical pharmacology studies are the basis for developing appropriate approaches for BE evaluation for long-acting injectables. For instance, the ClinPharm program generally gives you good information about the formulation release characteristics, the PK features of the long-acting injectable product for the reference product, and also the program generally provides a good information about patient features or covariates, if you want to call that may affect the pharmacokinetic features of the product. Also, it gives the information on whether the injection sites will potentially change the PK profile. And on top of that, it actually links the exposure change to the clinical outcome change through the exposure response relationship. That is a very important angle for us to consider. What are the key PK variables that we should consider for BE evaluation, and how do we really establish the relevant to BE criteria, and all the pieces of information can be put together to improve clinical BE study. Thanks.
Thank you for responding to that question. Moving on, we did just receive a few questions for Dr. Bing Li. And here is the first question for Dr. Li. What is the BABE criteria for the 90% confidence interval GMRs of Cmax and AUC?
Thank you for the question. I think your question is for BE, not BA. In general, if the 90% of confidence interval of the geometric mean ratio of test and the reference product for Cmax and AUC fall into 80.00 to 125.00%, then these two products are considered as bioequivalent. Some additional thoughts for you. If your product fall into a highly variable drug category or narrow therapeutic index drug category, then depending on the intra-subject variability of the reference product, you may end up with either a widened or a narrowed confidence interval. And for more details of these approach, please refer to the product-specific guidance for progesterone capsule. That was our model PSG for highly variable drug, and the product-specific guidance for warfarin. That is our model PSG for NTI drug product, which contains a stepwise procedure for statistic analysis, SAS code, and etc. for these type of products. Thank you.
Thank you for responding to that question. We do have another question for Dr. Li, and here's the question. Do we have different criteria for BABE and Cmax AUC?
Yeah, thank you for the question. In the bioequivalence arena, we normally apply the same 90% confidence interval approach for these two parameters. As we know, bioequivalence is defined as the active moiety of two products reach to its site of action with the same rate and extent. So these two parameters, Cmax being a reflective of rate of absorption and AUC for extent of absorption, are two important PK parameters to demonstrate BE. Thank you.
Thank you for responding to that question. Moving back up to the beginning of our panel, we have a couple more questions for Dr. Daping Kui. And here's the first question. It's difficult to recruit patients for BE study of antipsychotics. What are the other considerations to use a lead-in period to recruit patients who are not previously on the required regimen of the RLD?
Thank you very much for the question. Yes, so the product-specific guidance for some of the antipsychotic long-acting injectables recommend recruit only patient already on a stable regimen of the intended study product. So in general, it is not recommended to recruit patients on a different antipsychotic treatment and change the regimen solely for the purpose of enrollment in the BE study. However, to my understanding, this recommendation may still allow a possibility to implement a lead-in or a run-in period in the pivotal BE study. So this lead-in period is for patients who will be benefit from the treatment of the study drug, as per the discretion of the primary investigator. And in addition to that, the switching patient from other treatment, there should be some specific measures specified in the protocol, in the study protocol, to ensure there's no unnecessary harm to the subject. For example, the dosing, the running period should be consistent with ROD labeling. There should be information in the protocol on how these subjects are handled, including specific discontinuation strategy when applicable. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have another question in for Dr. Kevin Fang, and here is the question. Is it acceptable for virtual BE determination using AUC only?
Thank you for the question. Yeah, for the BE evaluation, we normally would looking at the older PK endpoints like the AUC and Cmax. So if you're using the AUC only, then you probably may missing the Cmax. It depends on the model. So you need to looking at and validate your model properly with the AUC and Cmax. Then you can do a proper virtual BE simulations. Thank you.
Thank you for responding to that question. We're going to move on to our next panelist. The next question is for Dr. Kandakar Alam. And here is the question. Verification of PBPK model seems like a huge task. Do you think the requirement of model verification to satisfy FDA may discourage the generic drug developers to adopt PBPK modeling? This is a moderator. Looks like we're still having a little bit of connectivity issue with Dr. Alam, and we're going to move on to the next panelist. And our next panelist that we have a question on is Dr. Sharon Thomas. And here is the question. Why is PUAC, why does it help ensure comparability during long-term treatment using long-acting injectables?
Thank you for the question. So as we discussed earlier today, partial AUC is recommended for single-dose B studies for several long-acting injectable. Now, the purpose here is to ensure that the exposure is comparable even after giving multiple doses. Long-acting injectables tend to have an extended treatment period, and we want to ensure comparability throughout the extended period. So the single-dose PK profile impacts the exposure even after the multiple dose administration. And hence we recommend the partial AUCs to ensure the similar PK shape. Let's take the example of octreotide. The single dose profile is characterized by multiple peaks here. The dosing interval for this formulation is only 28 days, but however, the drug levels will continue to increase even past 28 days. So in this case, equivalence in exposure during the time interval from 28 to 56 days will help ensure the similarity in the steady state profiles. Thank you.
Thank you for responding to that question. We're going to try to check back in with Dr. Alam to see if the internet congestion and connectivity issue is resolved. And we're going to try the question again that we just read out. And here is the question. Verification of the PBPK model seems like a huge task. Do you think the requirement of model verification to satisfy FDA may discourage the generic drug developers to adopt PBPK modeling? Dr. Lamas, as a moderator, I do apologize. We're not able to hear you at this time, so we do have one other question. We're going to move to another panelist and bring back up Dr. Sharon Thomas. Looks like we have time for one more question, and here is the question. What are the challenges expected in using partial AUC for long-acting injectable?
Thank you for the question. So some of the challenges that I can think of is we have to ensure that we have sufficient quantifiable samples that can be collected to enable adequate estimation of the partial area under the curve. Also, partial AUCs tend to have higher variability than the total AUC and the Cmax, and this may require a greater sample size. Given these challenges and other considerations, we do routinely evaluate if partial AUC should be recommended as a supporting evidence as part of our assessment process. Thank you.
Thank you for responding to that question, and thank you to all of our panelists for responding to numerous questions that came in. And we've just about run out of time. It gives us just a few moments to introduce, for closing remarks, Dr. Lei Zhang, who's the Deputy Director in the Office of Research and Standards in OGD. She was a member of the ICH Generic Drug Discussion Group, serving as the USA FDA topic leader. Additionally, she is a repertoire for ICH M13 informal working group that is developing the M13 guideline to harmonize bioequivalence study designed for immediate release oral dosage form drugs. Please join me in welcoming Dr. Zhang.
Thank you, Ray, for the nice introduction. Good afternoon, good evening, and good morning to all who participate across the world. I hope that you, like me, enjoyed every session of today's workshop. The breadth and the thoughtfulness of the information provided by our speakers, and also additional discussion and clarity provided by the speakers and also panelists at the question and answer sessions were enormous. This is the 5th year that we hold this workshop. We clearly see a continued interest from our stakeholders in this workshop, which is focused on the complex generic drug development and also other complicated issues that we are facing in generic drug development and approval. We have more than 2,500 people registered from more than 80 countries for this year's workshop. You probably saw the map shown by Brenda in the morning of today. Today, we covered emerging topics on the immunogenicity risk and also impurity assessment for developing generic peptide or oligonucleotide products. We also discussed about the comparative analysis involving device for the generic drug device combination products. That's quite challenging, and also future challenges we saw of developing generic drug products with electronic device and also prescription drug use related software. We want to prepare for those emerging challenges in the future. Also covered are biowaiver options, more efficient development approaches, new development of in vitro only approaches, BE approaches for injectable suspensions, and also you just heard the scientific advancement, including modeling and simulation to support development of generic long-acting injectable products. The Generic Drug User Fee Amendments, or GDUFA, started in 2012, have provided direct support for science and research. The scientific research funded by GDUFA program builds an essential bridge from the scientific knowledge of the modern era. Science and research are critical to modernizing the generic drug development program and the generic drug industry to continue to supply safe and effective generic drugs to the American public. With the expiration of the GDUFA II at the end of this month, we're also looking forward to the GDUFA III to be reauthorized. There's a number of enhancements for GDUFA III that are intended to further enhance efficiency, transparency, and gain more first cycle approvals. These include the PSG enhancement on new goals for newly approved complex products, options to request PSG-related teleconference and meetings, and also additional scientific meetings with industry, both prior to and post-submission, so that applicants can communicate with FDA in a timely manner for input that they needed for their development program to improve the efficiency and also enable better ANDA submissions. Enhancements also include the suitability petition process, including the establishment of new suitability petition goals beginning in FY 2024, and also new prioritization for the suitability petitions. Tomorrow, we will have a session highlighting the suitability petition and the changes that's going to be occurring in GDUFA III to maximize the use of the 505J pathway. Additionally, tomorrow we'll have another array of exciting topics, including additional complex products, such as locally acting GI products and nasal products. In addition, model-informed BE approaches will showcase how to support the generic drug development and approval. You will learn those new development in methodologies and those efficient alternative BE approaches to support complex generic drug development. In addition, you will also learn those common scientific issues and challenges we saw in generic drug development and also in generic drug applications, and also learn the practical considerations in study design and data evaluation that could facilitate generic drug development. As reflected by the title of this workshop, we try to better translate the complicated science behind generic drug development into more generic drug approvals. Finally, I would like to thank the session leads, speakers, panelists, and subject matter experts who worked diligently together to design and deliver the content for this two-day workshop. I also would like to thank our FDA Commissioner, Dr. Califf, for giving the keynote speech this morning. and SBI colleagues, Brenda, Ray, and others, in moderating the sessions and coordinating many behind-the-scene efforts. Thank you for your participation of Day 1 in this workshop. I look forward to your attendance of Day 2 of the workshop tomorrow. Thank you. Now back to you, Ray.